interested in using machine learning to solve all different kinds of biological questions. So I work on using both like artificial intelligence and the so-called coevolution to study protein-protein interactions. So before I start, I want to give you some general background about what it is, what is coevolution, for example. So if you study protein sequence alignments, and then so on the left, this is a protein sequence alignment. So in addition to those positions that are conserved, there's also pairs of positions where like the change in one position seems to influence the change in the other position. So these correlated positions in sequence alignment, they usually are caused by the fact that these two positions will come close to each other in the 3D space. So if two positions are close to each other in the 3D space, then they will influence each other in the course of evolution and result in this phenomena called coevolution. And if you do observe coevolution between positions, you can use this signal to predict which residue pairs would come close to each other in 3D space. And this is useful information for 3D structure prediction and for protein-protein interaction prediction. So, and also the methods for inferring such coevolution in protein sequences has been evolving rapidly during the last 2 decades. So between like 1990 to 2010, so the technique to infer this coevolution signal is still pretty simple. People are using some statistical method to look at one pair of residues at a time, just like what I was showing you just now, like manually. So that method, people can observe such a coevolution phenomena, but it's hard to use this phenomena to do any meaningful predictions. So then between like 2010 and 2017, there was a small breakthrough in the field of computational protein science when people used the more advanced statistical method, which is called global statistics. So these global statistics, instead of looking at one pair of residues at a time, it can be like analyzing all the residue pairs in a protein at a time. So that resulted in a significant improvement in the performance. So then people were able to, just from the multiple sequence alignment, predict what residues what residues will form contacts in the 3D space. So if you know like what residue would come close to each other in 3D space, then you would be able to predict the 3D structure of proteins. So at that moment, there was a breakthrough of using such a method to predict the accurate topology of proteins just from sequences. So, but of course, that breakthrough wasn't known beyond the people in our field until lately, where there's deep learning methods like RussellFold, AlphaFold that really was the most efficient way to analyze this coevolutionary signal. So this deep learning method, they are not only going to see the global picture of one protein or one protein pair, they also through the process of training, they already learned the rules from the entire protein universe. So they basically learned the rule of protein folding and interaction. So, and then as I showed that if you analyze the single protein sequence alignment, then by using the coevolution signal and the physical rules learned by artificial intelligence, you can very accurately predict the 3D structure of one protein. Let's say if you are interested in two proteins, then all you need to do is to look at the two multiple sequence alignment and see how they are co-evolving with each other. And if you use these two to analyze two protein sequence alignments, then you can get a coevolution, and you can use this coevolution to model protein complexes. Then how does it usually work? So this is a general workflow. Let's say you have two proteins, so it could be from a human or from a pathogen. Then you then you will find its like orthologs or its equivalent proteins in all other organisms. So it's either in other eukaryotes or in other bacteria. So you find the homologs of both your protein A and B. Then then you can assume if A and B would interact, then A, B probably also interact in several other organisms. So then what you would do, you would concatenate the protein A and B from each organism. And then you are converting this like two protein problem to a one protein problem. So once it's converted to one protein problem, then you can apply either the statistical methods or machine learning methods to identify the co-evolving residues in the concatenated sequence alignment. And for example, if you identify, you may identify many pairs of co-evolving residues. And usually for the case of interacting protein, these co-evolving residues, they would correspond to the residue contacts that will be formed on the protein-protein interface. So then you could try to see if these covalent residue will be compatible with a protein-protein interface. So that's no matter you use like a statistical method and do protein-protein docking, or you just use AI, which basically packed all these process all in a black box, but it is all basically doing the same thing. So it will just allow you to go from a multiple sequence alignment of two proteins to a protein complex. Then you can go even further, you can expand it and you try to repeat this kind of calculation for all possible protein pairs in the organism. Then with the assumption that if two proteins do interact, they will show stronger coevolution and they will come close to each other to form a meaningful protein complex. So if they don't interact, then you wouldn't see much coevolution. You also wouldn't see a meaningful complex when try to model them together. So then using by attempting to model all possible pairs, you can identify protein pairs that are more likely to interact. So that's what we do. We do such calculations on a protein-wide scale, and then convert this like a protein modeling problem to a problem of try to predict like in the organism which protein should interact with which proteins. So, but to do this like a protein-wide prediction of protein-protein interactions is not very easy. So first of all, it is computationally quite expensive. So let's take the case of E. coli. E. coli has more than 4,000 proteins. Most bacteria will have about like 4,000 proteins. That will mean 10 million protein pairs. So that's very expensive to compute. And another general problem of this protein-wide PPS screen is that it has a very low signal to noise ratio. So although you would have 10 million protein pairs, so you are only expecting to have like a 10,000 real protein-protein interaction. So that means for every true interactions, you will have about 1,000 like random pairs that you need to distinguish from. So then over the years, we've been working on this problem, and we were always like trying to develop different pipelines, but all these pipelines share a similar like design principle. So in order to cope with the scale of the calculation and be able to get the most accurate biological insights, we usually will design a hierarchical pipeline where we put the fast but not so accurate method first, and then using this fast but not so accurate method to identify a larger pool of candidate interactions, and then we will only apply the slower but more accurate method on protein pairs that are more likely to interact. So then when we just started, then this pipeline consists of like those local statistics and global statistics and 3D modeling. Then as we progress and as deep learning methods evolve, all these steps were replaced by different kinds of deep learning networks. So this is just a general workflow of how we do it. Let's say you can take a E. coli or any other pathogen, and then you can find each protein's orthologs in one array of other bacteria. Then you will generate the so-called paired protein sequence alignment, as I just showed you. You identify homologs for two proteins, and then you concatenate the proteins from the same organism. Then you can apply different either statistical or machine learning methods to predict the possible interactions. So, and then as you go through this funnel, you usually start with a rather large number, and then in each step you select the candidates that will reduce the number, but will keep the protein pairs that are most likely to interact. Then you go through many filters, then you will end up a relatively small set of predictions that we feel rather confident about. So then actually after So this was the work I did when I was a postdoc in the Baker lab. So then after that, I moved on to set up my own my independent lab, with the goal that we are going to extend this to eukaryotes. So, but however, it is a lot more challenging to make these things work in eukaryotes. So, because in bacteria, you can easily find like 80,000 different species, and you have a very rich coevolutionary signal from those many sequences. But in eukaryotes, you have a much fewer available proteins. So this make everything a lot harder. But the lucky thing is that the field is developing there's deep learning method. Although we are having a lot of weaker signal, but now we have much more sensitive methods to extract the coevolution signal from these shallower sequence alignments. So then we moved on to yeast. So which is like a model organism of protein-protein action, and it's a eukaryotes. So compared to E. coli, yeast has a little bit more proteins. So they form about 20 million protein pairs, and it is an established model organism for PPS study. So we can easily assemble two sites. One is like a positive control, consists of like real interactions, and there's also negative controls. They usually consist of negative pairs that show no evidence of interaction. So we in all our studies we try to make the negative control size about like 1,000 times larger than the positive control size, which reflect the true signal to noise ratio on a proteome-wide screen. So, and then I want to introduce one very important indicator of the accuracy for protein-wide PPI prediction, which is the precision and recall. So what are precisions and recalls? So let's say if you have a set of predicted protein-protein action, and you also have a set of true protein-protein actions, and then their overlap. So the better your method is, the bigger their overlap should be. So then if we use this overlap to divide by the total number of predictions, that is called precision. So that basically means what fraction of your prediction is right. So this is how clean your prediction is. And if you use this intersection to divide by the number of the total number of true predictions, then you get something called recall. This means like how comprehensive, how complete your predictions are. So these two metrics, they are usually reversely correlated. So then what we usually do is we will make like a so-called precision and recall curve. So we can make like a ranked list of predictions going from more confident to less confident. And then as you expand your number of predictions, usually your recall will increase and your precision will drop. If you have a totally random predictor, so then you will have a precision recall curve that look like this, a line that is very close to zero. If you have a perfect predictor, that means you will be able to rank all your true protein-protein action above the random pairs. Then you will get a curve like this. But realistically, you usually get a curve that is in between, and the area under the curve is a very good indicator of how well your method is working. I'm sorry about this technical detail, but this is really important for you to understand what we are doing in my later slides. So then as we start to uh, move from like our E. coli to yeast, the first thing we see is this global statistics, which is a method that works pretty well in E. coli, completely does not work. You can, as I said, like in this precision versus recall curve, the area under the curve reflects how well the method is work. So you can see it's really you can't predict much. So then, but at that moment, then AlphaFold came out, and then we also developed the Rosetta Fold, and then we also developed a fast version of Rosetta Fold that is designed for fast PPS screen. So then you can see this deep learning method, they immediately show a much better performance compared to the previous statistical methods. So then what we did is like, okay, then we apply this fast AI tool onto a large number of protein pairs from yeast, and from there we selected over 5,000 pairs that seem to have a higher chance of being true interaction. Then we applied AlphaFold to model the protein complexes for these more than 5,000 cases. And then AlphaFold is doing the modeling. It also helps us to select the protein pairs that does form a very good complex. So then if we apply a pipeline, we first use ReceptFold and then apply AlphaFold, then we were able to they being combined, you were able to achieve a precision recall curve like this. So what does this mean? So let's just focus on like one point in the curve. So when we set the precision to be 95%, then you can see the recall is about 28%. So this means like we can predict about 28% of all the interactions, and while making sure that 95% of our predictions are true. So it's just like, okay, we can predict a very confident set of interactions. We cannot predict all the interactions in the organism. So, but we can predict about like a quarter of all the real interaction in the organism without knowing anything. You just take the sequence alignments, then you already can know like a quarter of its possible interaction at a very, very high confidence. So then we just applied this pipeline to yeast, and then we were able to predict like more than 1,500 interactions. Then we were trying to analyze, okay, our predictions, do they agree with the previous knowledge? Then of course, they agree very well. Then about half of our prediction, they already uh, have experimental determined protein complex in the protein data bank, meaning that the facts they interact is known, and then how their protein complex look like is also known. There's also about half that, although the 3D structure of their complex is known, but the fact that they interact is supported by some previous experimental structure. There's a small subset of PPS that were not known before. But at that moment, so we were the first to show that AlphaFold can be used to model protein complexes and to model it on a on a large scale. And this modeling can reveal a lot of biological insights because now you have accurate 3D structure, then it will help you to infer the functions of protein, help you to understand how a protein complex would work. So that was a very significant piece of work, and also this just shows some of the protein complexes we were able to model, and then each of these complex has you can tell a biological story about it because it explains some mechanism of a protein function. I will just give you one example, which is this GPI transaminase. So the function of this protein complex is to attach the GPI anchors to proteins. So this is one kind of modification to proteins. So the components of this complex was known, but how these components come together and form a complex wasn't known. So this is our predicted structure back in 2021. So in the structure, you have like these 5 components. They come together and they form a complex, and there's a channel like in between that we suspect is the channel for substrates to go through. And right near the channel, there's a potential catalytic site. So shortly after our work, people also determined the structure of GPI transaminase, but it's after us. And you can see that the experimental structure agree pretty well with our prediction, and whatever we think is a substrate channel is indeed a substrate channel. So, and and then so at this moment, then we decide, oh yes, so all these deep learning methods seem to be working pretty well. Then we decided to, okay, using these deep learning methods to revisit the bacteria. So when we started this, we've only studied we mostly studied the E. coli, which is a model organism, just to like as a proof of concept. But now since we have better tool and we really can can confidently predict a reasonably large number of interactions from any organism. So then we decided to use these two to revisit the bacterial world, and we decided to work on a series of bacterial pathogens, and using a similar pipeline. So as outlined on the left. So we just took um, 19 bacteria of interest. Mostly these are human pathogens, so I listed all of them. And then we also found other bacterial proteomes, and we try to identify from which we try to identify and align orthologs. And then we do paired protein sequence alignments, and then we selected pairs with deeper sequence alignment. Because we have so many organisms to work on, we only want to work on protein pairs that are more promising. So then we still use our global statistics method just as a one pre-filter to select the more promising pairs. Then we use that Rosetta fold to so we use a version of Rosetta fold, which is a deep learning method, but it's a lot faster than AlphaFold. And then we use the AlphaFold. Then we follow the manual study. So again, we are laying like layering three different methods, and their accuracy is so each subsequent method is more accurate than previous one, but it is also a lot slower to compute. So we only apply these slower methods on a smaller set of more promising candidates. So, and then these are just some from these organisms, we did not really take all possible pair. So we decided to focus on proteins, on two kinds of proteins. So one is the essential genes, because if they are essential, that means if you inhibit their function, you can inhibit this bacteria, and that potentially can be a strategy to combat infectious disease. We are also interested in proteins that are known to play a role in pathogenicity or virulence. So we're also interested in identifying interaction to them. So like from each of them, we work on either essential genes or the known virulence factors and try to identify their interaction. So these are just the numbers. So like how many pairs did we decide to work on from each organism. So we totally put together like over about 5 million different pairs that went to the screen. Then so, and then yes, so here just a little bit detail about how did we did. So it's similar to what I introduced before. Like when you generate paired alignment, you always you start from one protein pair, then you find their homolog across different bacteria. Then you generate a so-called paired sequence alignment. And then another thing that I would highlight in this pipeline is this it's a new method that we developed. This is called like Rosetta Fold 2 Lite, which is a new method that we developed for this study, which is like another like a fast PPI screening network. So this just shows the so like a general idea of this tool. So to train this, so the architecture of this model similar to it's pretty similar to AlphaFold, except like for Rosetta Fold. So for AlphaFold, you have those evil former, which is a tension-based block that allow you to integrate the information between sequence alignment and the protein, and the interactions between residue pairs. But so in Rosetta Fold, we made the innovation that in addition to the sequence alignment and the pairwise feature, we also directly put the structure features into this evil former like block. So allow like some more free interaction between the sequence and pairwise feature with 3D structure features. So then when we train to train this model, we took like the known protein complexes from protein data bank. We used their structure. We also took like protein pairs that shouldn't interact, and as a negative control. So using both the positive and negative controls, we were training this network to predict the protein complex. And then, by the way, this network also learned to distinguish true interactions from false interactions. So, and then, so we apply this pipeline, then we identify thousands of putative interacting protein complexes. So here we just show a couple of example structures that we found from this work. And more importantly, we were always thinking, sure, we can make a lot of predictions, but it is hard to convince the experimental community that our prediction is right. Although we are very confident, we know like 90% of our prediction should be correct, but really to from whatever we predict to a like experimentally tested tested prediction, there's still a big step to go. So for this work, luckily we were able to recruit very capable collaborators who helped us to use experiment to validate some of our like unexpected predicted interactions. So this is just some experimental results that on two very novel predictions we made, and this is tested by bacterial to hybrids, and based on bacterial to hybrids results, then indeed that these predictions should be true. And we further expanded our experimental tests to use pull down assays. So and out of the five out of the oh yeah, so using pull down assay we tested 6 pairs, and 4 of them got like positive results, so they were shown to be interacting with pull down assays. Although one of them, although we did not detect the interaction, but we think this is indeed they should interact, but their interaction was just too weak to be detected by experiments. And one pair, like after further studying, is probably a false prediction. So this shows like the accuracy of our prediction is about like 80%. Yeah, so that was these were the tested positive case. This is a case that is a negative. There's one case, although not tested positive, but we think it's a more of an experimental problem. So then yeah, so that was the pathogen work. Then after that, then we're hoping like most excited about like the human interact. So then, however, we had a huge challenge to making all these methods work in human. So for example, this again, I'm showing this precision versus recall curve. This is this Rosetta fold, this new Rosetta fold method we developed for for the pathogen. But when you apply it to human, then its performance again become, well, not too good. So it put me into some like an imposter syndrome because people believe that we are the expert of predicting these interactions. But our methods, and most people care about humans, but our methods don't work too well on humans. So that was a difficult time, but we still, well, it's probably not really working too well for in general for humans, but maybe there are simpler tasks that we can work on. So the first thing that another thing we decided to work on is like, well, maybe we can work on the mitochondrial proteins of humans, because the mitochondria, one difficulty for all eukaryotes is you cannot get as deep sequence alignment as you can get for bacteria. But however, mitochondria is like widely conserved. Mitochondrial proteins is widely conserved in all eukaryotes, so you can get relatively deeper sequence alignment for mitochondrial proteins. So then we were able to just do a smaller scale analysis on human mitochondrial proteins, and we predicted some novel interactions, and some other experimental people reached out to us, and they were also able to validate some of our predicted novel interactions about like human mitochondrial proteins. And And actually, now we also made these methods work pretty well on human, thanks to my one of my postdoc who is extremely diligent and very dedicated to make it work. I we do have the so I am not planning to cover that work here, but just we are now making very exciting progress on human. I just want to show like again, precision recall curve is what we always monitor our performance. This is like before we seriously work on human. This is our like a mm, like precision recall curve. Then after a lot of heroic work did by my postdoc, then we were able to improve the method by about fourfold. So allowed us to make a lot of interesting discoveries in human too. And well, yeah, I do have a talk on that topic, but I think you guys will probably still a little more familiar with the bacterial work, so I will just stop here. And with that, I'd like to thank my own lab members and collaborators. So David Baker is my postdoc advisor. Yan is a leading author for the pathogen paper. He is a student that I worked. So although he was a grad student of David Baker, but throughout his grad school period, he was always working together with me. And then Aditya also another grad student in the Baker lab that worked with me, and Mingyan is my collaborator. She was also a postdoc in the Baker lab, so we together developed the Rosetta Fold, and I focused more on using Rosetta Fold for PPS screen. And then Joseph and Yassi, they are our experimental collaborator who helped us to validate some of our predictions in the bacterial pathogen, and another my collaborator who helped us to test our prediction about interactions in the human mitochondria. So yeah, that's all I have.
Thank you, Professor Kong, for the highly technical anticipated discussion.